Please stop making noise, Jen. All right, this is the nervous system, chapters 48 and part of 49. Neurons are nerve cells that transfer information within the body. They're organized into a brain in complex organisms, or in less complex, they might be organized into something called ganglia. Information processing occurs in 3 stages: sensory input, then the brain is part of the integration, and then there's a motor output. So somehow you sense information, our brain integrates it or makes sense of it, and then we have some kind of a motor output. One of the interesting things we're going to do in the next chapter is we're going to watch a video called Brain Games. There's a total of several episodes on Netflix, and they're really kind of cool because they talk about how this integration part is sometimes manipulated by your brain. Your brain perceives things a little bit different than maybe you actually sense them. The motor output is going to be part of your peripheral nervous system. Those are going to be all your nerves. And then the central nervous system is going to be the brain and the spinal cord. Brain and spinal cord is your central nervous. Those are the neurons that help carry out the integration. And then the peripheral nervous system is going to be the neurons that carry information into and out of the central nervous system. So those are going to be all your nerves. Sensory neurons are neurons that transmit information from the eyes and other senses to external stimuli or internal conditions. The sensory information is sent to the processing centers, so either the brain or the ganglia, and these neurons are going to interpret the sensory input. And then the motor output is going to rely on the neurons that step out of the processing centers, out of the brain and the spinal cord, and trigger a muscle or some kind of a glandular activity. So the neuron structure, and you move into this, is made up of a cell body, dendrites, axons, synapse, myelin sheath, and nodes of Landia. So here's 2 neurons. This is neuron 1 and this is neuron 2. So you've got your cell body right here. These are your dendrites. This is where sensory input is going to take in, or your stimulus is picked up. And then this is going to be your axon, and this is going to transmit the information to the synaptic terminal, which has got some dendrites here, and these are going to transmit the impulse into the next cell body, or the next dendrites of the next cell body, and that's going to submit a signal down to, again, the terminal and input. What you're seeing right here is most of the time one impulse is sent to the next neuron by neurotransmitters. These are chemicals that are basically sent from one across this synaptic cleft, or in this gap, sometimes as it's just called, from one neuron to signal the transmission of the other neuron. We're going to be focusing in class on this part here, the axon, and how a nerve impulse travels down that axon. These axons are also covered in things I'm not familiar. They're called myelin sheaths, and they are like this. And they are basically fatty substances that insulate the neuron, so it actually helps to transmit that signal a little bit faster. And they actually transmit like this. And so these gaps, these are the nodes of Ranvier. These are actually basically mist pieces. They travel across those. I think I've got a picture of that somewhere. Most animals have cells called glial cells, or glia, that support the neurons. Glia nourish neurons. They insulate the axons of the neurons, and they regulate the extracellular fluid around the neurons. So here's your neurons, here's your cell body and your axons. And then the glial cells are going to be right here, and they are just going to nourish and support those neurons. Resting potential, the inside of a neuron is negatively charged relative to the outside. So here's your neuron. I do terrible drawing. So this is a hollow area here. Okay, this is your axon. The inside of the neuron in here is going to be negatively charged in comparison to outside, which is positively charged. This charge difference, or what we call voltage, creates potential energy, and is called the membrane potential, because there's potential energy there to flip that. The membrane potential of a resting neuron is called the resting potential, and it is between -60 and -80 millivolts. The concentration of potassium is highest inside the cell, and the concentration of sodium is highest outside the cell and is maintained by the sodium-potassium pump. If you remember that from active transport, it requires ATP and pumps sodium and potassium across the membrane. The resting neuron has very few open sodium channels and more open potassium channels, which means that the outside of the cell has a higher positive charge, and thus the inside of the cell is more negative. So it's not that there's a lot more negative ions inside the cell, it just means that there is more positive on the outside. So here's that axon again. Remember, there's cell bodies down here and dendrites are down here. And so this is negative because there's more sodium out here than there is potassium. They're both positively charged, but because there's more positives out here, that makes this be negative. Here is a picture. So it's pulling a picture right from the dendrites. You could also pull this from the axon. You can pull from anywhere. Anywhere in the side of the cell. So we've got the sodium-potassium pump that helps maintain this. Remember, it moves 3 sodiums for every 2 potassiums. And then you've got a potassium channel and a sodium channel, and those move only potassium or sodium. And so you'll see that up here, sodium is really high. And then down here, potassium is really high. And so potassium is going to be moved this way, sodium is going to be moved this way. That's with their gradient, so that does not require energy. But the pump is going to go just the opposite. It's actually going to move potassium this way and sodium this way. So it's actually moving it and creating this gradient. That's part of what creates that gradient is the sodium-potassium bond. The membrane potential changes in response to a stimuli. Neurons contain gated ion channels which open or close in response to stimuli, which alters the membrane's permeability to particular ions, which in turn alters the membrane potential. Hyperpolarization is where it makes the inside of the membrane more negative, which results from any stimulus that increases the outflow of positive ions or the inflow of negative ions. So this is where you really are at resting, is where you have the more negative on the inside and the more positive on the outside. Depolarization is a reduction in the magnitude of membrane potential in which a stimulus causes the gated sodium channels in a resting neuron to open and sodium diffuses into the cell. So remember, sodium now is going to diffuse into the cell, and that's actually going to flip or reverse this. So we were kind of here, now we're going to be here. We're going to move to a reversal of that. That's called depolarization, and then when it flips back, that's called hyperpolarization. So here's just some graphs to kind of explain the stimulus. Here's your action potential. It's triggered by a depolarization that reaches a threshold. So remember, here's your resting potential. It can increase, but if for some reason it doesn't reach the required amount of membrane potential, the amount of difference isn't great enough, it's going to just fizzle out. But if the difference is great enough, it'll actually trigger a depolarization, a hyperpolarization, and then back to the resting potential. You'll see here, this depolarization did not actually produce enough didn't meet that threshold, and so it didn't create an action potential. I kind of like to put an analogy to this, like a toilet. This is actually what my high school psychology teacher did, and it worked pretty well. It stuck in my brain, at least. The threshold is like if you're flushing a toilet, if you don't hit that handle hard enough, it won't hit a threshold and it won't flush the toilet. But if you hit it hard enough, it will actually flush the toilet, refill the toilet, and bring it back to its resting potential. If depolarization shifts the membrane potential sufficiently, that's where we got to reach that threshold. The result is an action potential, which is a massive change in membrane voltage. Action potentials spread along the axon, making them well suited for transmitting a signal over a long distance. Action potentials occur when the membrane voltage is increased to a particular value called the threshold. It's about negative 55 in mammals. Action potentials are an all or nothing response. That's where I get back to the toilet. You can't like halfway flush the toilet. You either flush it or you don't. Depolarization opens the voltage-gated sodium channels, causing further depolarization. And there's a positive feedback because we're working towards a goal, which triggers an action potential whenever the membrane reaches the threshold voltage difference. So here is where we're at in kind of stages here. The resting potential, you have more positive outside, more negative on the inside. And this is actually, there will be another one over here of the same. This would be another membrane here. And it would be negative here and positive out here. Some people get confused with that. This is actually the membrane. So if you're looking at a cell, here's your body and your dendrites, and then your axon and your terminal. This here is actually two membranes. It's a membrane here and then a membrane here. So we're only showing you the one membrane right there. So you've got your sodium channel and your potassium channel. It's moving, it just naturally is moving sodium out and potassium in. We have a depolarization where all of a sudden we're moving sodium in and we're moving potassium in, so we're rushing everything kind of in. And then that's going to flip, this is going to keep happening and it's going to go along that axon. Eventually, it's going to be enough of an influx in that it's going to flip those charges. So it's going to be enough influx to create now a positive charge in here and then a negative charge out here. It's going to move enough of it. And then when that happens, it's going to move along and then it's going to go back and reverse that. So now we're going to go back to moving the sodium, back to moving the potassium, and now we're going to go back to a positive here and a negative on the inside. And as you're going along an axon, one part of the axon here will be like this, and then back here behind it, it will start to repolarize. So this is showing a graph of that in terms of the membrane potential. So we've got 1 is your resting potential, 2, you're meeting that threshold, you're getting there, right? You're starting to move those ions, but then you hit that threshold, and all of a sudden now you flipped the membrane voltage, and then we're going to fall back down, we're going to hyperpolarize, and that actually comes usually when that dip hits down below that threshold. And then we're back at, we have an undershoot where we're really low, and then we get back to our resting. And that's just, again, that diagram in a little bit larger view. The refractory period is a downtime between before another action potential can be initiated. For most neurons, it's active only about 1 to 2 milliseconds for the onset of an action potential and the end of the refractory period. So we don't even notice that. The frequency in which a neuron generates action potentials varies in response to the input. For example, in hearing louder sounds results in a more frequent action potential in neurons connecting the ear to the brain. So this is showing what I was talking about before, where we're having the action potential here, and then as the action potential moves down, we're going to repolarize here, and then the action potential, and then we're going to repolarize here behind it. So it's going to move along that way. Mutations in genes that encode ion channel proteins can cause disorders affecting the nerves, muscles, brain, and heart. It depends largely on where in the body the gene for the ion channel is expressed. Mutations affecting voltage-gated sodium channels in skeletal muscles can cause myotonia, which is periodic spasming of the muscles. So basically overexciting, not supposed to be doing it when it's not supposed to be. Mutations affecting the sodium channels in the brain can cause epilepsy. You know what epilepsy is, like having seizures, kind of fall asleep in the seizures. The evolution of the axon structure. Evolutionary adaptation enables faster conduction of axon insulation. The myelin sheaths around the vertebrae axons, insulating them and speeding up conduction. Gaps between the myelin sheaths are called nodes of Ranvier, which is where the voltage-gated sodium channels are located. It means the action potential jumps from one to the other, speeding up the process. I was backwards on that, sorry, in the first diagram, so back. Oh, I missed, how did I miss this? Oh, because I'm going too fast. And those, I was talking about how it jumps from one to the, it jumps from one gap to the other, one node to the other. Sorry, that was my bad. So it's going to jump in between there. The myelin sheath, it's going to jump the gap here and skip over those myelin sheaths. Sorry, that was my bad. I use these for these. Oh, there we go. So here's the picture of that. So here's the nodes of Randy are here, and that's where it's going to jump that gap. And this is a myelin sheath that's going to insulate it. It's going to help to move the signal along a little bit faster. Think about like why your wires are insulated in your house. And that shows it again. Here's where those channels are going to be. Synapses can be electrical or chemical. Most of them are chemical. Majority are chemical synapses, which cause the release of neurotransmitters across the synapse. The presynaptic neuron, that's going to be the first neuron, packages the neurotransmitter into a vesicle, and then it sends it to the postsynaptic neuron. The release from the presynaptic neuron is the result of calcium that is released in the area. It can either cause excitatory or inhibitory effects on the postsynaptic neuron. So it can either cause something to happen or it can inhibit something from happening. A lot of what you're going to learn about part of your mass addiction activity is looking at how certain drugs actually mimic neurotransmitters and how that messes with your brain and how that causes the addictiveness of drugs and also the symptoms of drugs. So here's your two neurons. This is your presynaptic and this is your post. Now, when it moves on to the next one, this will then become the pre, this will become the post. So we're just moving that along. And so where the terminal end meets the dendrites is where that's going to actually happen. These are the neurotransmitters. Calcium influx is going to cause these neurotransmitters to be packaged into vesicles and open that up and release them into this synaptic cleft or gap, sometimes it's called. And then those are going to be picked up by gated channels on the postsynaptic synaptic cell. And then that's going to then become your presynaptic, and it's going to go along the next one, and then on and on and on. Researchers have identified more than 100 neurotransmitters belonging to 5 acetylcholine amino acids, biogenic amines, neuropeptides, and gases. This just looks at the structure of a lot of those. Things you might have heard of before, dopamine, serotonin, those are your feel-gooders. Vital for the nervous system functions that include muscle stimulation, memory formation, and learning. This is acetylcholine. It's a group of neurotransmitters, not one, but they can cause a signal to be sent, or they can inhibit a signal. The amino acids, glutamate is the most common and has an excitatory effect on the postsynaptic neuron, so it causes something to happen and to send the signal. The key role in the formation of long-term memory. GABA, most inhibitory synapses, and increases the membrane permeability to chloride. Diazepam, which is Valium, is actually a artificial one that reduces anxiety through binding to a site on the GABA receptor, and so it's going to cause an inhibitory effect with the GABA, and that's going to help so that people that have anxiety or something are going to not have that excitatory. Dopamine and serotonin these are biogenic amines. Dopamine and serotonin are released from many sites in the brain in sleep, food, attention, and learning. Some psychoactive drugs like LSD produce their hallucinatory effects by binding to the brain receptors for the neurotransmitters. So then that way, dopamine and serotonin aren't able to bind because the drug is actually binding to that site instead, and that's what causes a lot of those. I did not mean to do that. I think I actually kept all my things. I didn't want to do that. Neuropeptides, endorphins, you've probably heard about those too. Natural analgesics, decreasing pain perception. If you get hurt, usually endorphins kind of like take over, and so you don't feel pain a lot of times early on. They're produced in the brain during times of physical or emotional stress, such as childbirth, and that's probably why we choose giving birth, even though it hurts. In addition to decreasing pain, they also decrease urine output, depress respiration, and produce euphoria. So even though you might be in pain, you may not feel that pain. Opiates are drugs that mimic endorphins by binding to the same receptors and produce many of the same psychological effects. Nitric oxide released during sexual arousal contains certain neurons in human males release nitric oxide into erectile tissue in the penis. In response, the smooth muscle cells in the blood vessels walls of the erectile tissue relax, which causes the blood vessels to dilate and fill the spongy erectile tissue with blood, producing the erection. Viagra actually works to increase the ability to achieve and maintain erection by inhibiting an enzyme that terminates the action of the nitric oxide. Stuff you ever knew. Learn in biology. That's the end of that one.